Okay, I think we can get started. So today for this session, we're so happy to welcome two pioneers in the field of culture meat. We have Dr. Anne-Claire, who is from Bit.Bio, the coding company, and Benjamina, CEO of HireSticks. So I'll let Anne-Claire take it off. Please take it away.
Okay, so let me share my PowerPoint. Yes. Okay. Hello all. Nice to meet you. Today I'm gonna present you what we are doing in the lab. My background is in stem cell biology. I did my PhD in France, in Évry. I was working in cardiac differentiation of embryonic stem cell from 2008 to 2012. And I was working on the very rare disease, and I did my postdoc on the same rare disease, but on the adipose tissue side of this disease, where I become an expert in adipose tissue biology. And then I've been recruited in Cambridge in a very big adipose tissue biology lab of Antonio Vidal-Puig in the Institute of Metabolic Science. And I joined then Bit.Bio. So I'm an expert in mesodermal differentiation in general. And today I will introduce you to some of the stem cell in general and pluripotent stem cell in particular. So just a brief introduction about pluripotent stem cell, their history and the different type of stem cell. I don't know if you are familiar with that or not. Then the Bit.Bio technology, what is cellular programming, and the optioic technologies that we are using in the lab, and some application of this technology, and of course, making clean meat with animal pluripotent stem cell. Let me know if you don't understand anything or if you want me to explain more or to slow down. So the history of stem cell is that in 1963, Till and McCullough demonstrate the existence of hematopoietic stem cell, and it was like the first stem cell. So basically, they took some irradiated mice, they put some cell in it, some hematopoietic stem cell, and they were able to make other cell. And it was a pioneer in embryonic stem cells. And in 1981, Kaufman and Evans derived the first mouse embryonic stem cell from the mouse embryo. And for that, they obtained in 2007 the Nobel Prize of Physiology and Medicine. In 1998, Thomson and collaborator isolated the first human embryonic stem cell, which was a big step in the field of pluripotent stem cell. And in 2006, Yamanaka's group induced the first mouse fibroblast into pluripotent stem cell called induced pluripotent stem cell, and they get the Nobel Prize in 2012 for that. And in 2007, actually, he derived the first human cell line of induced pluripotent stem cell. And this is a very important discovery for the company I'm working in, because basically our company is based on this reprogramming. And in 2013, Mitalipov group, they first derived a human embryonic stem cell for somatic nuclear cell transfer. So it's another important discovery in the field of stem cell. And basically, I'm talking about stem cell, but there is different kind of stem cell. So you have the totipotent stem cell, which is the product of the fertilization, and it generates absolutely every cell in the body, including the placenta and the extraembryonic tissue, and it can form the entire human being. Then, so this is the most potent stem cell. Then you have the pluripotent stem cell that generate every cell in the body, except placenta and extraembryonic tissue, so they cannot form an entire human being. And then you have multipotent stem cells that are really specific. They may or may not have plasticity. It depends on the type of stem cell. They are really useful, but you cannot do all the type of cell with that. And what is pluripotency then? So pluripotency is a transitory state of embryonic cell or induced pluripotent stem cells that exist only during a brief window of development. It's like it's a very artifactual actually to have them in a dish, because it's in development, it doesn't last more than 3 days in humans. And even during these 3 days, it is always remodeling all the time. So you have like, so pluripotent steps. You have the naive, the prime, the intermediate progenitor. Not everyone is still okay with what's the definition of pluripotency. There is a lot of discussion around that, and that's why it's fascinating for me. And in theory, the stem cells that are deriving from the embryo, they can be maintained indefinitely in the pluripotent state in vitro, as well as now induced pluripotent stem cell. And the pluripotent state is a continuum of states. That's why it's so difficult to cultivate this state, because it's really an artifact. But the thing that is wonderful about this cell is that in theory, you can reproduce them indefinitely. In practical, it's not so true, because then they can have a chromosomic aberration. But this is why this is so fascinating also, is that because you can do basically a lot of things with just one vial of cells. And there is two types of pluripotent stem cell. You have the human embryonic stem cell arising from fecundation, going through the morula, then the blastocyst stage. In the blastocyst stage here, that you can see here, you have the inner cell mass, and this inner cell mass, you can derive it and obtain embryonic stem cell. Or you can take some cells that are already existing in human, like fibroblasts. This is the cells that are the most used in the field of human induced pluripotent stem cell. And you can put these 4 factors that are called the Yamanaka factor or Thomson factor. This one are actually a mix of both. And so NANOG, OCT4, SOX2, and SEMIC, and induce some somatic cell into pluripotent stem cell. And as a result, you have both pluripotent stem cell with the advantage and the inconvenience. The induced pluripotent stem cell now is better, but there is some reprogramming that is maybe not complete comparing to embryonic stem cell, but then embryonic stem cell are the disadvantage with ethical problem. And what we are doing in Bit.Bio is that we take this pluripotent stem cell and we are doing reprogramming, but in the other way. So we take some transcription factor involving cell differentiation, and we do differentiate itself from this pluripotent stem cell. And we are doing that thanks to OptioX. And OptioX, it's enabled and control a scalable reprogramming of stem cell. So basically, you take your human embryonic stem cell, your human iPS cells or animal iPS cells. And then with that, what is OptioX is you have the TET responsive element with your gene of interest on one genomic safe harbor, and on the other one, you have RTTA. And what's happened is that when you put docs, when you don't put docs, you can cultivate your cell indefinitely. They're still pluripotent like normal cell, except that they have this machinery in them. But when you put docs, you can differentiate this cell. And it has been published in 2007 with the academic laboratory of Mark Cotter, that you can differentiate these cells into neurons, glutamatergic neurons, oligodendrocyte, but also skeletal muscle, thanks to this, setup and this OptioX system. And that is really nice, because the biology now is entering into the cell reprogramming era. It's not only now reprogramming somatic cell to pluripotent stem cell that has been initiated by Yamanaka in 2007, but it's also differentiated cells from any pluripotent stem cell or multipotent stem cell. You can, it's been shown that you can put transcription factor in a cell and obtain your cell type of interest. There is a lot of reprogramming in the literature. Most of them come from fibroblast sources, but you can do that also with induced pluripotent stem cell mainly. And the main target cells so far are neuron and cardio, but there is also adipocyte, beta cell, hepatocyte, myocyte, blood cell. I mean, you can imagine any cell type that you want. And this is really, really nice. So what we are doing at Bit.Bio, we are using this cellular reprogramming to do basic research, or to do normal and disease cell model and normal cell model for developmental and cell biology. We do cell for drug discovery, so also a normal and disease model for drug screening, target discovery or toxicology. And we do also, there is a plan to do cell therapy with a different cell type. And the thing that is interesting you today is also bioproduction. So we can do specialized cell type because thanks to the technology of Bit.Bio, these cells are really highly scalable. So why we need to do clean meat? Because of course, I mean, you know why, but just as a reminder, because meat production is really resource intensive and the use of antibiotic can create antibiotic resistance in human, and intensive animal culture can also create pandemic. And we really well know that now. And the advantage of the cell-derived meat is antibiotic-free, it saves water, it's animal slaughter-free, and it requires approximately 90% less resources than classical meat production. So you have also, so you have different way of doing clean meat. You have the classic way of doing clean meat that the most of the protocol for doing clean meat arise from. So, sorry, I have a problem with my pointer. Yeah, sorry. Up. Yeah. So the classical clean meat production, I mean, at the beginning, all the pioneer in clean meat production, like Mark Post and all, is like you take a muscle progenitor cell and or a supporting cell and or endothelial cell and or adipose progenitor cell, and you differentiate this cell and you can do clean meat in vitro, which have their advantage too. But the only problem of this kind of doing this, and it can be overcome at some point, I don't know, but there is this problem of the batch-to-batch variability and a difficulty in upscaling the cell production. And so the advantage of the Optioic system is the scalability once again. So one of our mission in Bit.Bio is to make an animal cell for clean meat industry. So for that, we need to establish the best culture condition for porcine pluripotent stem cell, generate porcine cell to overexpress muscle and adipose transcription factor for myocyte and adipose differentiation respectively, and also establish the best culture condition for porcine, this time differentiation into myocyte and adipocyte. So the hurdle to overcome are scalability, consistency, price, and taste. And we need to implement as pioneer in the field, the cell quality, the robust differentiation, the media development, and the cell mass production. And Optioics, the advantage is that it solves some of the hurdle, not everything, but some of the hurdle. At least you solve partially scalability and consistency with this technique, because when you do directed differentiation with growth factor from pluripotent stem cell, it's expensive because you have to pay a lot of cytokine. The protocol are not really easy, they are not really consistent, they are not really scalable, and you don't have a good purity, maturity. And I developed protocol for a long time, so I really agree with that. But the advantage of reprogramming is that you will gain into purity, maturity, scalability, and consistency. So basically, when you put the docs, you already obtain a cell that is differentiated and is really consistent. So we use this Optioic technology with porcine pluripotent stem cell to obtain muscle and adipocyte. So for muscle, we use the same principle of media that we use for the iomyocytes that we are using with human. These iomyocytes are like a product that is ready today post-revival, and you can do like very nice myocyte with that are multinucleated. They are able also to reply to acetylcholine, so they are functional, and you can see that there is like multiple nuclei. And this is a video that I really like and that convinced me to work in Bit.Bio actually, is that when you start the differentiation, you can see that in like 8 days, you have muscle fibers that are forming, which is for, so this is on human cells, which is for a stem cell biologist that I am. I know how difficult it is to do muscle cell. And like Olivier's Pourquier protocol is taking on average 60 days. So it's quite crazy that we are managing to obtain so fast some myocytes. And it's working very well. I'm cultivating them from time to time, and it's really working well. So basically, I just wanted to show you that because we use this technology and we apply this technology to to porcine pluripotent stem cells. So basically, we have porcine pluripotent stem cell. We did the genetic engineering with muscle transcription factor. So we obtain a porcine pluripotent stem cell with optioix muscle. So when you cultivate them, they look like normal iPS cell. And when we add the docs, we do the transcription factor induction, you can see that we obtain muscle cells with very nice striated and multinucleated. But we don't only did muscle cell, we did also adipocyte cell. Why? Because adipocytes are responsible for the palatability of the meat. They have actually, it's really complicated, the different type of adipocyte that you can have in an animal. In pork, there is like, and beef, there is like different type of adipocyte. You can see that you have plenty of adipose tissues that don't have the same function of insulation, or the taste will not be the same. This is not exactly the same type of adipose tissues. This is really complicated. But the problem of that is that there are like very few protocols, and they are difficult to obtain, and very little is known about adipose development. But thankfully, we have the ability to do adipocyte differentiation in-house. So for that, we use the same cells, and we genetically engineer them with adipocyte transcription factor to obtain optioix adipocyte cells that also look like the pluripotent stem cell. And when we differentiate them, so we induce the transcription factor of interest, you can see that we have adipocyte cells. So all in all, using the optiox technology, we have generated myocyte and adipocytes that are scalable, of high purity and consistent. And the thing which is super cool is that it has finally unlocked precise and scalable manufacturing pork and cell for meat. And I would like to thank you for your attention. And I would like particularly to thank you, the team that is working with me, Sarah, Maddie, and Patrick, because it's a lot of work to generate this cell, and all the lab, of course. And I will be happy to take any questions that you might have.
Oh, thank you so much, Anne-Claire, for this very insightful talk. It was very nice. So we'll have more Q&A. If you can save your questions for the end, but I want to let Benjamina from Higher State go with her presentation. Thanks.
Yeah, let me just share my screen. 1 second. Check. Can you see my screen?
Yes.
The one with just like the, great. 1 second. Great. So hi, everyone. I'm Benjamina. I'm the founder and CEO of Higher Stakes. So I'll skip to some of it because it was already explained before, but I'll spend a little bit more time towards the end of my presentation through how we can bring up open innovation to cultivated meat, which is the subject of the day. So as was mentioned, one of the key reasons why we need cultivated meat is because of some of the big challenges caused by the meat industry. But we decided to focus particularly on pork because of several reasons. Partly, it's the most widely consumed meat in the world. Also, there is a huge amount of antibiotic use. So there's as many medically important antibiotics used on pigs in the US than on humans, which is crazy to think about. There's also a lot of different processed products that we can start with. And at the moment, a massive shortage with African swine fever. And so there's been a massive rise in alternative proteins, but people still like the taste of meat. And when we surveyed consumers, we still found that people were unwilling to remove meat from their diet, and most meat eaters actually prefer to move to cultivated meat versus plant-based meats. So huge market. I'll skip on that. We use iPSCs as well that you just got explained, so similar on that. We're starting with pork belly and bacon specifically. So pork belly is more aimed at the Asian market and bacon more at the European and US market. So I'll spend most of my time on this slide so that you don't get things that are repeated from both presentations. So I think one of the challenges is how do we have open innovation, but at the same time, companies, need to keep their IP and need to keep things protected. And so how do you mix things together and make sure that, we still have some things that are shared? And I think one of them is enabling technologies. And while, yes, we've seen more and more companies creating enabling technologies, so things such as media, things such as scaffolding, there is also a lot of opportunities for things that are really just open source and used by everyone. I mean, one of the ones that to me kind of jumps out is even just basal media. It's a pain to filter. You can get all the elements themselves really, really cheaply, but to actually have a company do it and mix everything in-house and all of that is really, really a pain. And so people can still, there is opportunity for people to do that for everyone. There's a lot of technologies that today are still really, really at the start and are way, even within our own moonshot, which is cultivated meat, are still too much of a moonshot. Examples, how do we make sure that companies are properly accredited and can prove that they're cultivated meat to the consumers? How do we make sure that on the long term, when we have, a premium for being cultivated meat, how do we make sure that meat consumers, meat companies don't pretend that they're cultivated meat? How do we make sure that we can show, this is higher stakes or this is Bit.Bio's meat, and things like that are, things that the companies themselves is way too much of a reach, but there is still a lot that can be done. Creating cell lines for maybe even more exotic animals. You have one company called Vow that is really focused on exotic animals, but again, there might be room for more. There's so many little problems that can enable the whole industry, and that just the companies is not at the top of their priority. Around consumer acceptance, this is something where really we need to work together, and the more we're open about it, the more we work all together, the better it is. If we start going in the media and saying Bit.Bio are horrific for the consumers, it's not going to benefit us. We need to all work together and make sure how can we deliver a message that puts the whole industry forward. Now, each company can have their own marketing and can have small things that differentiate them, but putting other companies down won't help the whole sector. We need to move the whole sector forward. So anything that we can do to innovate and work all together in terms of consumer acceptance is definitely a big one. When it comes to safety, I think that's one that's maybe that's very important, that people have underestimated how much work I think there will be, what are really the new standards that we'll need to set, what are the new challenges that we'll need to set. And again, having people, and when it comes to safety, the more we can show that it's open and that everyone adheres to the standards, the better it is, right? So when we submit the novel food regulations, which is an 18 months process in Europe. And the more we can point to open data, the more we can point to studies that have not been made by our company and that are external, the more we can prove, hey, it's not just us that are biased that are showing this. There's groups that are completely independent working on this. The more there are groups showing how we can better monitor our process, how we can better assess whether a product is safe, what are the standards that we can set, all of those things are extremely important. And we're seeing some of the NGOs start and do this and get everyone together, but there's not enough. And I think having today, a product that's on the market in Singapore and having all of these things not yet set is a big risk. And we really need to start pushing on some of those safety aspects. And what can we do? What devices can you create? Things where you can innovate. And, so you mentioned biophysics, informatics, you need to have different people coming together and taking on from different industries and putting it all together. Another big problem is if we're going to save a lot of land, what are we going to do with all that land? What can we do to make sure that that land is used for things that advance us and don't counteract? If suddenly we free up a lot of land and all of those go into factories to make things that are going to pollute the world, that's not going to help. So we really need to think about some of those bigger challenges. Things like packaging, again, how can we create packaging that's good for our meats? How can we make sure that those packaging are sustainable? There's so many tangent challenges. How can we ensure that cultivated meat use the most renewable energies when they create their manufacturing? There's so many new things that as a company we think about, but not always aren't able to put all our full energy into it. And I mean, Clarice and I did a couple of years ago a hackathon in the space, and it was quite fun as well to see like the different ideas and the different things that people came up with. And I really just think we need more of that. And yes, companies will always have as a priority to protect their IP, and it's very difficult to get companies to necessarily share. But I think I see as well a lot of companies going towards a little bit of a Tesla model where people will file patents, but also allow other people to use those patents just so that we can advance the whole industry. But obviously still patenting it enables people to have that control. And so if they ever want to remove it, they can. So that's me. It's a little bit shorter because I skipped some of the parts, but we're also hiring if any of you are interested, and I'm happy to answer any questions. Thank you very much.
Well, thanks to both of you for this amazing presentation. I'm just stepping in to say that every time after these kinds of events, we stay on this call for around half an hour to discuss the next events for Biology Club. So if you want to stick around after, you're welcome. And yeah, if anyone has any question for either Benjamina or Anne-Claire, please, we are listening. Or if someone is not comfortable in turning on their microphone, you can also ask your question on the chat and we will mediate after that.
Yeah, so I suppose, thanks very much for the talk, guys. It was really interesting. I suppose one of the questions that I'd have in terms of the cultivation of meat and such is like, obviously within the pharmaceutical industry, we look at this is a certain dose, this is set, you can't go beyond these limits, your upper and lower limits. With meat, does that become a small bit more skewed or kind of more gray in terms of how much you can put in, the ratio of meat or the ratio of meat to fat and such? Do those variations play a factor in, I suppose, your kind of production process? Do you allow for a certain amount of fat over, say, a certain amount of meat to get where I'm going? When you're kind of shopping for meat, you'll always see some people would tend towards the more fattier meat, or some people would tend to more leaner meat. Is that something you take into account when producing?
Yeah, so I can start, and Claire, feel free to add. We do take into consideration, but I mean, the main thing also is we could create a bacon that's fat-free, but that's not really going to be the best, the best taste. So it's a mixture between having the variable muscle and fat percentages, but also creating a product first and foremost that consumers want and tastes great. But yes, I mean, again, on the long term, we do see having different percentages, and we can control this a lot better with cultivated meat.
Perfect, thank you.
So for me, it's different because we are generating the cell in Bit.Bio, but we are not doing the meat. So I don't really know. I just know that it's really complicated, and actually the standard for cultivated meat are quite close to the pharmaceutical industry. It's really in Europe. It's really strictly followed. So, but I don't know what are the percentage. But I guess as Benjamina was saying, also you can maybe play with that depending on the consumer.
Yeah, and they haven't yet told us as well whether there is a percentage needed of cultivated meat. So there's different companies have different percentages. So some have like 10% cultivated meat, others have like 90%. And what can you call cultivated meat? Because obviously on the meat side, there are strict rules on what can you call meat if you have a product that is 90%, something else. For example, plant-based or starched, and yeah.
Yeah, you answered that perfectly. I was wondering on the kind of the variance aspect there. It's like, to me, it kind of looks like you've kind of a double variance stage, like within the experimental process itself, and then with the resulting the results of that process. It seems like a kind of double variance, where you have the variance within the experimental phase and the experiments of the results then, or the variance of the results then after that. Yeah, perfect. Thank you.
I have a question about the, as I understand it, you're going from the stem cells to chunks or layers of meat. How do you? But no blood vessels. So how do you actually keep your culture alive during the process?
I can't disclose.
Once again, we are generating the cell, so this is not one of our interrogation.
So there must be some method by which you keep it alive. I was just wondering how you scale it up to an industrial level.
I think that sorry, go ahead, Benja.
No, go ahead.
I think that it depends on the product. So if you want to have structured meats, then might be more difficult. But for them, while doing more processed meats, then they can have like perfusion bioreactors, or just bioreactors that like better delivery of the nutrients, like continuous flowing through as the cells grow, which mimics kind of the blood vessels in the body.
So you're not actually chunks of meat or layers of meat. It's
right, it's more processed.
Individual cells. Okay.
Yeah, cell biomass. But they can make structured meats. It's just the technology is much more complicated there, I think.
Yeah. The other thing is you will still be you will also be subject to bacteria, possible bacteria contamination and virus contamination. So in spite of your comments about not using antibiotics, I think you might end up using antibiotics.
So far we haven't used antibiotics, and so we managed to culture our cells without antibiotics, and you can do this in closed systems. Obviously, all of this is extremely controlled, especially when it comes to the really large volumes that we will have to do.
Okay.
Same here, we cultivate our cell without antibiotics.
Okay.
Hey, thanks for your talk. So having read about this, it seems that the FBS seems to be one of like the biggest cost factors. And I was wondering if you are working towards being able to do this without FBS, or if you think it's possible.
Yeah, I mean, I think both of us, and correct me, Anne-Claire, if I'm wrong, but I think both of us have, one of the advantages of working with iPSCs is that they grow really well without FBS. they still cost a lot. There's still a lot of challenges with them, but FBS is not one of them.
Thanks.
Pleasure.
I think we got similar questions relating to this antibiotic resistance and the FBS. So Somalia asked if the meat derived from tissue culture would contain a lot of chemical and undergo lots of lab process. So wouldn't it affect human health?
Well, this is why we have regulations in place, and to make sure that, everything is controlled and that everything that could be harmful is completely removed. And again, the regulatory approval won't pass if anyone uses things that aren't safe for humans. That's the whole point of passing through a regulatory approval, is that human health, it proves that that safety is number one. And if we look as well at what's happening in the meat sector, there's a lot of things that are going on that are not necessarily healthy for human beings. But obviously our aim is to be really healthy and to not have anything in our processes that could hurt anyone.
Yeah, I agree with that. Also, the thing which is quite easy with forward programming is that you don't need so much cytokine, in your cell culture. So it's like it's really easier.
Just a follow-up question on that. So if there's regulations, and then I'm supposed there'll be a lot of regulations before the product comes out, is there any new category that you envision that will develop? Because is it going to fall completely into food level regulations, or it is going to be going into a drug level regulations, or is there anything for the future that you would envision that the new lab grown meat would have a separate regulation of their own?
I don't see why it would. So it goes under the novel food regulations, which is for novel foods. There will be standards that will be expected, I think, from cultivated meat companies. But having a completely separate regulatory system for cultivated meat, I don't see, at least in the near future.
Yeah, I have absolutely no idea about that. Sorry.
Hi there. Can I just ask a question about manufacturing, just so I can get it in my head? Right. So let's assume we're manufacturing this stuff at scale, and I don't even know if that is done at this point. But what are the fundamental inputs? If you're manufacturing this at scale, I assume there's energy that needs to go in, but there's also some forms of raw materials. Can you just talk about what that even looks like? I don't have a handle on that.
In a very broad sense, because obviously can't go into details for, I know
no,
this.
No, but I mean, it's just like fundamentals.
But so it depends as well on the growth, or whether you talk about a growth or the differentiation stage. But overall, you have your cells, you have your media, which is a mixture of vitamins, potentially growth factors, depending on whether you use growth factors or not. For example, Bit.Bio doesn't.
No, I don't say that we don't use growth factor.
Sorry about that. Yeah. You can, in some methods, not use any growth factors, or you use growth factors, vitamins, sugars. So your standard kind of media, if you think about cells, I mean, and manufacturing, you're really, you're growing and you're differentiating cells. You have the scaffold that's coming in when you differentiate the cells. I'm not sure how much more I can tell you than what's been said in the presentations, to be honest.
Okay. No, I don't have a handle on you. If you're running this at scale and properly generating like the levels of meat that at a world scale, what I'm just trying to work out, what do those raw materials, as the whole production line before you get to the meat thing, what's that look like? Is it fields and fields of sugar beet, or is it You know, is it, what is it? I don't even know. Like, you know, so if you're creating aluminium cans, you put in aluminium ore and crap loads of electricity, but that's very different to if you're creating something else. I'm just trying to get a handle on what does the supply chain before the proprietary meat factory look like? What's, you know, if you're doing washing powder, you need a lot of chalk and a little bit of magic to, you know, that's fundamentally what you sell. So what is the impact?
I mean, if you
and I don't mean proprietary, I just mean like the fundamentals.
I mean, I guess as Benjamina was saying, it's a lot of media, and it's a kind of if you look at the cell therapy as an example, you need like a large cell culture volume, so it could look like this.
Okay. And is anyone doing any of that sort of manufacturing at any scale at this point in time, either in a parallel industry that could just be switched on, or is this like from scratch, like doing it at this scale versus cell therapy is, you know, 10,000 times more you'd need, and therefore this is effectively creating a whole new pre-supply industry?
Depends what you count as scale. I think there are several companies that kind of medium scale, and there's one, I think, that's really actually preparing for scale. But okay. No one's, you know,
yeah, I mean,
working millions of kilograms, no one's done that yet.
Okay, yeah, and I was meaning the raw materials that would be needed for that, because it's the whole supply chain before it as well. Right?
The media.
Okay, yeah, interesting. All right, sorry, thank you.
I have a question there. Do you envision something like, you see, having everybody culturing their own meat in their own home, sort of a 3D printer kind of thing? It's not a 3D printer, but you know what 3D printer done is, instead of taking it from industrialized manufacturing, you can have it to your home, making your own prototypes. And so could we think about in future, say 5 years to 10 years down the line, somewhere you have a small bioreactor at your home, that's your actual cooker, and you have these raw materials like things getting in, and the chefs get creative to make their own recipes for cultured meat? Any comment on that?
I think you need to be really sterile, and I wouldn't cultivate cell in my own home. So maybe there will be system at some point that will, I mean, I don't know what the future is made of, but I wouldn't like to cultivate my cell in my home anyway. I prefer to be in a safe class 2 safe cabinet. So, I mean, maybe, I don't know, but I think also in terms of cost, I'm not sure it's worth it.
Right, yeah.
Yeah, and I guess it depends as well in what way it's made. Yeah, it's made, because in some way, if the cell is not already kind of a muscle or fat, you still have the time of differentiating, and people can barely think about what they want to eat, you know.
Yeah, maybe home is a bit overstretched there, but even if a restaurant, a chef wants to do something.
Yeah, I think again, I can see on the long term, on the really long term, maybe, you know, maybe having mini factories in supermarkets or restaurants. I think home is a little bit of a stretch, and I think it's, again, we'll have to see whether that makes sense from an economies of scale perspective. The way it looks like today, it doesn't. But who knows what the future holds? And I know that's the aim of some companies that are out there today.
Yeah, so I have another question there, coming back to, I suppose, the kind of a regulation question, I suppose, or a comparison kind of standpoint. When you're looking at, say, producing likes of pork belly, say per cube, are you also looking at kind of a reference standard in terms of what's already out there in China? Say, you know, per cube it's 6 grams of protein, 9 grams of fat, maybe 0 grams of carbohydrates, and then adding to that, are you also able to control it from a micronutrient perspective? Are you able to, you know, we see in the likes of cereal industry at the moment, you know, the fortification of some cereals through the addition of vitamins and such. Are you looking at that kind of down the line, maybe, you know, from maybe not, I suppose, a therapeutic standpoint, but maybe to add a bit more value to what you're already producing?
It's something we're aware of, but I can't comment further than that.
Yeah, that's fair. No problem. Thanks.
I have another question if nobody else has. Just thinking, do you think one of the reasons that we don't have culture meet yet is because there's so much kind of overlapping because of the secrecy between all the different companies? There's a lot of overlapping innovations just being repeated as everyone has to start from zero instead of building up on open innovations.
I don't think it's the reason, no. Would we have gone faster and would there be more innovation? Sure, absolutely. On the condition that there would have been a same amount of funding if everyone had been open source. So if you live in a world where everyone would have gotten the same amount of funding all together as a big group, again, I don't think we would be today producing 1 billion kilogram. I think we would be further than we are. But the reality is, if it had been open source and everyone had been to work together, there would be less incentive to fund some companies, and then there would be less innovation. And so it's a little bit, and that's why this debate is so tricky. But yes, obviously putting more minds to it usually results in more, but you also have some very strong characters that all have to work together and all manage a project that is way bigger than if you have small startups. So the real world is not as simple as, yes, if we all put it together, it would be there today.
Interesting. You don't think the jump is that big just for any startup to overcome? It's actually managing like perfect balance.
I mean, there is a jump. There is, and again, it depends when you started. There's been definitely an acceleration, and there are things at the same time that now are easier than they used to be. So I just think the world is a lot more complex than that, and without creating a proper model, and it's very, very hard to say. And even then, it would be very, very hard to predict what would have happened. But I do believe strongly that, private companies can push things a lot more forward. Now, can we maybe at some point benefit from acquisitions and mergers? Absolutely. But I do strongly think that, private companies are going to be the ones pushing the industry the furthest. But I'm biased.
I think also the pluripotent stem cell in our case is a very recent field. So I don't think it's, I think there is so many questions that it's also interesting to have different point of view on this to generate different things. So I don't think in this case, it's just nascent and there is everything to invent. So I think for the moment, I think it's actually quite interesting to have this too, because then everybody will try to find their solution.
No, that's a great point, because I think if we had all, let's say, followed Mark post, no one would be using iPSCs. Everyone would have tried to keep optimizing, adult cells, and we would have lost some great innovation. So again, strongly believe competition can lead to great innovation.
I see, thank you. Is there any final questions?
I suppose one more. Sorry, now I'm piping in with a load, but I know, obviously, secrecy is obviously warranted. It's a business. It's at the end of the day, and especially it happens in science all the time. No one's going to just throw their research out the window and go, "Oh, here, look, this is what I discovered," because it's just going to kick themselves in the foot. But do you think, when this is brought to market, obviously, currently, we like to think that we can look on the back of a packet and see what we're going to eat, in terms of micronutrients or macronutrients. Is that secrecy going to be maintained into the marketing aspect? Are you going to be able to put on the back of a packet, look, these are the vitamins within, these are the macronutrients, or will that slightly give away your position on how it's produced?
Not really. You know, when people take chicken and put it in bleach and then take it out and give it to you, there's no, it's not written bleach on the package. So, you know, obviously that's not the example we want to copy, but it's just to say like the process, there's the processing part of it, and then there's what you put on the package is what's left, right? And so what we'll put on the package is what's left. And yes, if your whole IP is what you think is special, is having a new type of, you know, vitamin included, great. But, you know, yeah, I mean, again, if you're going to have a vitamin included that's not usually in meat, you're probably going to want to advertise that anyways. And so I expect it to be on the package.
Yes, perfect. Thank you.
I strongly agree with that. It's a matter of process. So you can have the recipe, the final recipe, but if you don't know how to do it, you will never manage to do it.
And the amounts, right? Like look at Beyond Meat, you know what's in it, but that doesn't mean you can create the Beyond Meat tomorrow.
And I also think that spending most of my life in academia, I'm not sure that actually it's so different than in companies, because you want to publish and then you have to return information and you still have very much innovation in academia. So you need just to find the balance. And actually, I don't think it's so much different. I agree that open innovation is really nice when you can do it, but when you cannot, I'm not sure it's so problematic.
Well, I think you want a mixture, right? There are some things that should be open and some things that shouldn't be. And I think you get the best out of both worlds when you mix. And the things that should be open, again, I think things that are safety are really there's a strong argument for having that really open and shared. A lot of things can be open, but not everything should be.
Perfect, thank you. Do you envisage in the very, very future now, when this all gets going, that maybe there is a chance that you could market your own type of meat under your own company name? Is that something that you look at for the future? You know, that you're not just doing pork belly or like bacon strips. Do you envisage doing your own kind of company meat, like a brand, like I suppose a branded meat, for lack of a better word?
Yeah, we do.
Very good. Thank you.
That could be very cool. In terms of a bio lab, a bio makespace, is there, and this is a question for anyone, I guess, is there equipment that if it was available to people, obviously it may be capped at £100,000 worth of equipment, not £1 million worth of equipment. But if we had £100,000 to spend on equipment to put in a bio lab, what might we put in there that would be interesting to people to enable them to do things they couldn't otherwise do? And nothing is fine, because that's cheaper.
I mean, that it depends of which type of cell you want to use at the start. It depends of the amount of money you have to cultivate these cells, because then you will choose carefully which type of cell you want to use. So
where is your question get? Like, what are you trying to get to? Like, are you?
So I guess the question is, you know, we have a limited bio makespace in Cambridge. What facilities would it be great to have in Cambridge that could help this industry, either as amateurs or bringing people together, or whatever it could be, that perhaps otherwise would be trapped within one company and therefore not get the collaboration, or wouldn't be available in Cambridge?
I would say going through it right now, but it's like not going to cost you 100K. Having a space that has a pilot, labs for cell culture and kitchen, development kitchen space all together.
That's funny, because one of the things I've always wanted to add to Makespace is a proper Heston Blumenthal style kitchen.
But yeah,
not obviously that's not quite what you're thinking in your mind. But yeah.
Good job.
Makespace has kind of a kitchen.
Sorry?
Makespace has kind of a kitchen, but also I think we're working, I think a good place to put this out there, that the Synthetic Biology Society is working on an open source bioreactor, like automated bioreactor. So if you want to join the project.
Well, we had quite a lot of interest in sourdough starters. I think some of the chefs around Cambridge are quite interested in this, some of the stuff, some of the ones I've been talking to. So yeah, that, Benjamina, what you're talking about there, that's kind of what I'm getting at. I don't know what the answer is, but there's some people who know a lot more about this than I do, who might have.
We do have a chat on that. I mean, it's a problem that we're going through now, so happy to help.
Sure, yeah. Yeah, well, yeah, you don't need to tell me, just chat in the biology club, yeah. Cool.
Does cooking parameters come into play here too? I mean, obviously, you're producing to produce something that's a likeness to a certain meat. Does there come an experimental phase in the kitchen? Because obviously they don't come out of the bioreactor or whatever process, just cooked. So is there some sort of is there also an experimental phase within the kitchen in terms of what kind of taste you want to get?
Yeah, absolutely. I mean, obviously the process, I mean, an example, I tried some vegan sushi the other day. And as you do, I decided to think, oh, well, how is it going to act when I cook it? And I cooked it and it completely melted. And, that's not what we want. Yeah. So, we need something that looks, that looks good in the different shapes. And this is more or less important for different cuts and different meats. It's more important if you're doing salmon and if you're doing a sausage, and it's partly pre-cooked, but yeah, it's definitely important and definitely part of the process.
Perfect, thank you.
Okay, there are no more questions. Thanks so much again to our wonderful speakers. And for this very lively discussion. I invite you to join our channel, so the biology group, Google group, and then keep up with the next event. So we have how to build it in the Makespace by Abby soon. So yeah, keep up on the meetup and join us on Mattermost. Also, if you're interested in getting involved in some of the groups, we group projects. So as I was talking about the open source bioreactor, or if you want to talk more about potential events we can run, etc. That would be nice to see you there. And yeah, if you want to stay after this to discuss how to get involved in biology group, please do. Thank you so much again, and Dr. Anne-Claire and Benjamina for this talk.
Thank you.
Thank you very much. Thank you very much for inviting.
Yeah, really interesting. Good stuff.
Bye.
Thank you. Have a nice evening.
Evening.
Thank you.
Bye.
Bye.